Good afternoon, everyone. I am Dr. Domini Devi from University of Malaya, and I'll be chairing this session. Our first invited speaker for this session is Associate Professor Dr. Leslie Tiang Lun Tan from University Putra, Malaysia. He'll be presenting on the topic Molecular Diagnostic in Medical Mycology, Better Late Than Never. Let me introduce a little bit about Dr. Leslie. Dr. Leslie Tian is an associate professor in the Department of Medical Microbiology of Faculty of Medicine and Health Sciences, University Putra Malaysia, UPM. He is also involved in the Diagnostic Service of Microbiology Lab in Hospital Pengajar UPM, also known as the UPM Teaching Hospital. His area of expertise or research includes medical mycology, fungal diagnostic, and pathogenesis and probiotics. He has published over 60 research articles in peer-reviewed high-impact journals with close to 1,000 citations and H-index of 16. He has also written several book chapters. He holds several intellectual properties and serves as an academic editor in Biomedical Research International Journal. He is currently the honorary treasurer for the Malaysian Society of Lactic Acid Bacteria, a member of the Malaysian Mycology Interest Group, which is under the Malaysian Society of Infectious Diseases and Chemotherapy, and is also a member of the International Society of Human and Animal Mycology. Without further ado, I would like to welcome Dr. Leslie to deliver his presentation. Dr. Leslie, the floor is all yours.
And can you see my slides?
Yes, Prof.
Okay, all right. Thank you again to the Madam Chairperson, Dr. Damani, and for your kind introduction. So good afternoon to all participants. First of all, I'd like to take this opportunity to thank the organizing committee for giving me this opportunity to present in this very esteemed conference of yours. So again, I'd like to introduce the title. My title is the Molecular Diagnostics in Medical Mycology, Better Late Than Never. So give me a few minutes to introduce my campus. My campus is, I'm from UPM. UPM is known as the Green Campus of Malaysia, and we are the second best public university in Malaysia. And here is where, on the bottom left, is the picture of the faculty where I work, and also next to it is the picture of the newly built hospital that we have, or Hospital Pengajar UPM. So I also like to take this opportunity to extend to all participants, if you intend to do your postgraduate degree, you might want to consider UPM. UPM currently stands at 132nd position throughout the whole world, according to QS World University Ranking. So here are the contents of my presentation. First of all, I will be sharing with you on fungal diseases, what are the spectrum, what are the laboratory methods that we currently have, the advantages and disadvantages, and why we need to have an accurate diagnosis, especially not just to know that it is fungal infection, but we also need to know the species because of the reasons that I'll be sharing later on. After that, I'll be sharing with you on the molecular diagnostics of the medical mycology, what are the platforms, the types, and also, of course, the different advantages as well as the disadvantages in each of these types. Okay, followed by the issues and challenges in molecular diagnostics for medical mycology, and I will end my presentation with the future of molecular diagnostic in medical mycology. So basically, this whole presentation is to convince you that to adopt molecular diagnostic as part of your diagnostic tools in addressing fungal diseases. So a little bit about fungal diseases is basically, it is a spectrum ranging from the superficial nail infection, skin infection, to the invasive type. Okay, here are some of the photos of fungal infection. You see here the nail infection, ringworm, and also the vagina candidiasis But the one that actually catches the most attention is the spectrum where it involves the fungal disease that affect people with weakened immune system. Among them is listed down there. So here are the pictures of the invasive aspergillosis. Here is the causative agent, the Aspergillus species. And not long ago, I think, I do not know what's the number of cases now, we also have black fungi, which catches the attention of many people, happening in India, certain region of India, where the presentation of the name of the fungal disease is mucormycosis, where the clinical presentation always showed that necrotic tissues that happen around the rhinocerebral region. So this is how the different spectrum of fungal diseases, and of course they have other eye infection and also foot infection and all that. So what do we have currently as our laboratory methods? So most labs, I believe the participants here will be very familiar with the conventional methods. You have the direct microscopy, and you have the histopathology to support, and also you cannot run away from culture, which is the gold standard of medical mycology thus far. And we also have serology, where we either detect the antigen of the fungus and also the antibody produced by the host as a result of the infection by the fungus. Okay, here are some of them. And I think not many of you have serology, access to serology tests too for fungal diseases. And needless to say, molecular methods, I think, from the very short survey that I did, I can safely say that throughout whole Malaysia, I think that there may be less than 5 labs throughout the whole country that actually carry out molecular methods for fungal diseases. So here is the table showing you that different approaches in approaching fungal diseases and the different diseases, and also the different approaches. Basically, what you can sum up from this table is various approaches have their strengths and weaknesses. So the take-home message from this table is basically that you cannot just rely solely on one method to come up with a good diagnosis, but you have to have at least 2 or more to come up with a good diagnosis. So a little bit about the epidemiology of the fungal diseases. On the left-hand side, you can see that this is the fungal diseases, and on your right-hand side is the mortality rate. Basically, this slide trying to tell you that fungal diseases has a very high mortality percentage or mortality rate. Take for example, invasive aspergillosis. The mortality rate can range from 30 to 95, depending on 2 factors. Basically, the first one is the early diagnosis, and secondly is whether you give the right antifungal to the patients. So if none of these 2 are fulfilled, the chances of mortality is very high, approaching 100, meaning to say that out of 10 patients that you have down with fungal diseases, 9 or all of them will die as a result of these fungal diseases. Of course, you have other spectrum of fungal diseases which are also very high in their mortality rate. So this slide basically wants to tell you that it is very hard to actually just rely on one method as mentioned previously. This is histopathology of fungal tissues isolated from patient. To an expert eye, to mycologists as well as to histopathologists, yes, it is quite easy to tell that there is fungal elements present in the tissue section. And if you look further, you can see that there is a hyphae, okay, with a cross-wall receptor and with a 45-degree branching. So this is so much that histopathology can tell you. To tell you what species they are, it is still so much work to be done, okay? Yes, for picture A is, yes, it is Aspergillus, but for picture B, with the information that you know, like 45-degree branching of the hyphae, you will tend to think that this also belongs to the hyaline mold. But in fact, this is not Aspergillus or nothing near Aspergillus. This is actually Rhizopus, which is one of the member in the Mycology genera. So these things can happen if you just rely on a single platform to come up with diagnosis. And also for picture C and D, confusion can occur. You can easily see that this one can looks like a ribbon-like hyphae. Immediately you will think that it is Mucorales member, but in fact it turned out to be Aspergillus niger. So yeah, the basic take-home message for this slide is that you cannot rely on a single method to come out with a good diagnosis for fungal diseases. So to add in more complication to these fungal diseases diagnosis is basically that the signs and symptoms for fungal diseases are non-specific. It's quite similar to all the other bacterial infection and also viral infection. Colonization is sometimes very difficult to distinguish from invasive disease. Okay? Blood cultures are commonly negative. I think most of you may have already experienced it. Close to 50%, up to 50% are culture positive. For invasive candidiasis, for invasive aspergillosis is even worse. Patients are often unable to undergo invasive diagnostic procedures to retrieve valuable specimens for diagnosis, like bronchoscopy. Culture and microscopic examination still remain the gold standard, as mentioned previously. But you know, and I know that they are insensitive. Antigen assay, yes, it has its plus point, but it varies in their sensitivity and specificity too. Okay, when it comes to molecular method, yes, most of the molecular method in-house, we have yet to clinically validate these in-house methods. So this is one of the challenges which I will deliberate later in the following slides. So why is it important to actually have an accurate diagnosis for fungal diseases? Because we are seeing increase in prevalence simply because the number of immunosuppressed and immunocompromised patients are in the increasing trend. Mortality is high, and also lately we are seeing the emergence of antifungal resistance, especially on Candida glabrata, where it is we can see resistance against azoles as well as the echinocandins, antifungal drugs. We also have Candida auris that are multi-resistant, and also Aspergillus fumigatus, which are more and more isolated to have and to show pan-azole resistance. Basically, this table is trying to tell you that it is very important to get the ID of the specific organism because these different organisms respond differently to antifungals that we currently have. And we also like to just share with you that antifungals are very toxic, especially to the kidney. If it's given to the wrong patient, it will actually make the condition of the patient worsen. So what are molecular diagnostics currently in medical mycology? So basically, molecular diagnostic, similarly to all the other disciplines, is basically looking into detecting the DNA or RNA or any of the gene products of an organism. And for this matter, is the fungal species. So most of the time when we talk about molecular diagnostics, we cannot run away from polymerase chain reaction, which is PCR. So the current approaches in approaching molecular diagnostic for medical mycology is basically two. First is we come from broad aspects which we can utilize the pan-fungal PCR assay, where this pan-fungal PCR assay can be good in the sense that it can tell the attending clinicians that whether a patient is sick, is it because of fungi or not fungi? So it helps to detect all fungi, so-called. But as explained previously, we cannot just stop there by knowing it is a fungi, where we need to know the genus, and also if we would like to know the species of the fungus to arrive with a good diagnosis. So what are some of the types that we currently have? PCR, okay, with its different formats, conventional PCR, nested, semi-nested PCR, real-time PCR, isothermal PCR. Conventional PCR is very straightforward. I think most of the participants may have already known PCR has been popularized by COVID because PCR is currently now the gold standard in diagnosing COVID-19 cases. Nested semi-nested PCR is basically an extension of the conventional PCR to increase the sensitivity of the detection of PCR in the events that we have low number of cells for fungus, and also especially those formalin fixed paraffin embedded tissues where it's, the amount of DNA extracted can be very low. Real-time PCR is the one that is currently used for COVID. Similarly, we can utilize this platform for fungal diagnosis. Isothermal PCR is the one that is coming up. Some examples are like LAMP, NASPAR, and rolling circle amplification. LAMP basically is low amplified mediated, basically all these three processes happens at an isothermal temperature, meaning to say that you don't really need PCR machine to carry out these processes. Okay, so that is the added advantage of this isothermal PCR. Besides PCR, you have nucleic acid hybridization probes. You can harness the use of fluorescent in situ hybridization of fish. So what you have is a probe with tagged with a nucleic acid sequence that is specific for certain species. And in that probe also, you will have this fluorophore that is tagged together with it. So a little bit of a manipulation on the clinical specimens that you need to do basically is to release the DNA that is from the cell or the tissue, so that when you hype it later with the probe, you will be able to let the probe bind to the DNA of the particular fungus, and you can view them under fluorescent microscope. And the third one is the magnetic resonance, and this is the latest FDA-approved device to diagnose invasive candidiasis. So this is the sole device approved so far for invasive candidiasis. So here is a picture of comparing between two, the conventional culture method, which can take up to 7 days, depending on how fast the fungus grow, and sometimes it doesn't even grow. And for PCR, if you have all the facilities within your setting, you will probably be able to complete it within a day or two, if not maybe 3 days, if you need to send, for example, the sequencing that you get from your PCR to outsource to other companies which offers sequencing services. So that will take up a little more time. So basically, these are the process that you need to go through when you do a molecular diagnostic for PCR. You extract the DNA, you do the first round PCR, you can run it on the gel, and you can, if let's say it needs to be further amplified, you can do semi-nested PCR to further amplify the product that you get from the first round PCR. And from there, you can send it, the PCR product for sequencing. Okay, and of course, from sequencing, you need to use this sequence, and then you need to go into gene bank of the NCBI gene bank to find out which sequence is homologous to the sequence that you have. And from there, you will be able to identify the fungal species. So what are the common target genes that you use for PCR in medical mycology for molecular diagnostic? Is the ribosomal rRNA genes, as well as the internal transcript spacer gene. So why these two genes are used? Simply because it has this conserved region where you can design primers, and these primers can bind to this region, and this region is conserved among all the different fungi species. Okay, so the advantages is if you use these primers that from this conserved region, you'll be able to amplify many fungi, if not all the fungi. And so that's the reason why these two particular genes are selected for the PCR in molecular diagnostic in medical mycology. So besides that too, you also have other genes like listed down here, okay? And because that two genes may not be suitable for some of the fungal species, or if you want to do further speciation, you can also use this gene as for you to design primer from. Why do we need molecular diagnostic in medical mycology? So these are the reasons that I can think of. It's because mortality is high, and I know that, and I'm sure some of you may agree with me that it has a shorter turnaround time, and time is always very key, especially in patients with fungal diseases, because the later you administer the antifungal drugs, the worse the patient can become. okay? Growing yeast or fungi that cause infection is a very problematic issue, especially rare molds that are very resistant to antifungals. So molecular diagnostic is very flexible in that sense, unlike the other laboratory methods that you have. And existing methods lack sensitive specificity and sensitivity. And we all must acknowledge that the number of mycologists around are not many, and not many of us are interested in taking up mycology simply because it's not as sexy as virology as well as bacteriology. So that is also one of the reason why we need to have a molecular diagnostic to support the diagnostic service in our lab. And also, of course, the training programs right now is quite hard to come by. So that adds another reason why we need molecular diagnostic in medical mycology. This slide is a busy slide. Basically, it's just to tell you that it takes 20 years for Aspergillus PCR to be accepted as one of the criteria to arrive and to diagnose invasive aspergillosis. So take heart, maybe one day Candida PCR and also other fungal diseases PCR can be adopted as well. So some of the issues that I can think of, first is of course sample preparation. It is not as straightforward as bacteria and a virus because cell wall of the fungus are hard to break. But with a little bit of knowledge and with a little bit of effort, I think this will not be a big issue. False positivity, yes, we know that fungal molds are present all over the places. So we need to be very careful in our interpretation. The issue with true infection, contamination, or colonization is also another one. The next issues is we need slightly higher technical skills and requirements needed for molecular diagnostics. I think thanks to COVID-19, I think all our skills are much better now with regards to molecular lab testing. If you opt for commercial kits, they are expensive and may not be cost effective depending on each of your lab setting. And if you have in-house assays, we may have some issues with MDA because this reagent may not be MDA approved. And cost. Everyone is talking about sustainability. Yes, we always have to keep in mind about cost. If we would like to do some analysis or studies in looking into cost effectiveness of using molecular diagnostics as part of the methods in our lab. Okay, if you use DNA sequencing, you need to be aware that gene bank may have some sequence that are not properly curated. So yeah, so you need to be aware of that. Sorry. So how to overcome? We can have validation of in-house assays on its accuracy, precision, and clinical utility, proper interpretation, together a collaboration with the clinical microbiologists and clinician, and so that we can arrive, not just that if you get a positive result, meaning to say that it could be the pathogen or the fungus that caused the infection, having in mind that contamination or all that can happen. We need to establish fungal burden in respective settings to know that this is normal, this is not normal if you get certain fungal species. And we need to refer to properly curated gene database for sequence comparisons. So don't just simply dump into NCBI. And as explained just now, the problem with regards to NCBI gene bank. And for all of us here, since most of us have not started any molecular diagnostic service for medical mycology, my advice is we can start small, but we consistently and persistently do offer this service because I see big potential in molecular diagnostic for medical mycology. And of course, you can also collaborate with labs with molecular facilities if you don't have one in your own setting. So here is just the table showing you the strengths and weaknesses. So of course, in as much as I'm interested to introduce this method to you, you also need to know that it comes with weaknesses too for some of this method. Okay, PCR sequencing, whole genome sequence is coming up. hybridization, mass spectrometry, which some of you may have the access to multi-TOF. Here are some of the commercially available molecular assays. Some of you may be familiar with that, but may not have the access to it. I think it boils down to cost. Okay, you have the T2 BioFire, Septifast, life cycle, and so on and so forth. They utilize different methods with their advantages and limitations. So what do I see in the future of molecular diagnostic for medical mycology? I think that point of care testing will become the in thing. Perhaps for a start, maybe it will not be done bedside yet. Specimen will still be sent to the lab for careful testing. And I must thank, not to say that I like to have COVID around. I think nowadays most of our laboratory staff are equipped now or more trained in molecular works, and that is an advantage to start the molecular diagnostic for medical mycology. And we also have more equipped lab with all the centrifuge, with all the PCRs machine and all that. So thanks to COVID, but no thanks, shoot, COVID. For next generation sequencing is the next one. There are some labs throughout the world that are trying to use next generation sequencing, perhaps maybe not in any future, not in future for our country because of cost, but definitely it is something that to look out for. And of course, with the advancement of sequencing technology, we have the 4th generation sequencing technology that use nanopore technology to generate sequencing based on detecting changes in electrical conductivity. So here is a workflow on the NGS setting. So similarly, you get consultation, specimen, extraction, library preparation, sequencing, data analysis, bioinformatics, and also in the end, what you get is a phylogenetic classification. You have all the different fungus, and you also need to have a bit of intelligence in bioinformatics and also looking at all the clinical history to interpret the results. Not all that are present in the report are the pathogens. So here are some of the publications that I have. We previously utilized semi-nested PCR to detect 10 medically important Candida species. And we also utilize real-time PCR for Candida species differentiation. And that work resulted in us submitting for a patent, and the patent is accepted. And recently, my PhD students did a review on comparing the LAMP technique in comparison to conventional PCR techniques. You may be interested to read. And this is very fresh from the oven. This is on the modified and improved DNA extraction method for molecular detection of Candida species from positive blood culture bottles. So this actually shortened the turnaround time because normally you will need to have pure culture to do, and then that will take up another 1.5 days to do the PCR. So take-home message. So I hope I can convince you that actually molecular diagnostic in medical mycology is something that is very important. So we need to establish molecular lab for fungal diseases simply because of the reasons that I shared. Secondly is for those who have already had experience in molecular diagnosis, you need to share your experience with us so that we can properly strategize. And as you know, molecular diagnostic may not be sustainable if you do it solo, okay? And again, if you have already some facilities in your lab, start with what you have in your lab. So I will be very happy to share what I know and with you with regards to molecular diagnostics. And again, the title of my presentation is Never Late to Start the Molecular Diagnostics. So it's better late than never. So here I'd like to acknowledge a few individuals. I'd like to thank my supervisors previously, Professor Dr. Xiao Heng Fong, and the late Professor Dr. Ng Ki Peng from UM. He taught me mycology. And also Professor Dr. Chong Pei Pei, who is now in Taylor's. He was my co-supervisors. And my collaborator, Dr. Rukumani Devi Velayatun from also PPUM. And also my students in mycology lab UPM, and also my colleagues in my department for all the support and rendered in my work. So I'd like to end my presentation by this poster. When you think of fungus, you actually save lives. And wishing all Malaysian salam kemerdekaan, and thank you for your attention.
Right. Thank you very much, Prof. Leslie. But due to the interest of time, it's already 2:30. We have to move to the next speaker. If any of you have any questions